Okay, so we can start the first session of this afternoon, who will be given by Igor Aronson, and will present collective effects of active particles using phase field models.
Okay, thanks. So if you think that phase field theory for individual cell migration is simple, you're probably right, because now it will be something really complicated. So I will talk about phase field description for multiple cells. So usually cells don't act alone, and our body has about 37 trillion of cells. And the question is, how do they interact? And one of the most common interaction is just excluded volume interaction. It's shown here. So if you have multiple cells, they just don't penetrate into each other, but they can deform each other. So they keep going, but they exert forces, but excluded volume interaction is the most common. So depending on the concentration of the cells, they can form some collectively moving clusters, as shown here. So individual cells move more or less randomly, but most cells, they develop adhesive contacts and develop some tissue clusters, and they move together. So we've been working on it for quite some time, and we developed probably the first, we developed a description how to describe multiple cells moving using phase field. And the idea is the following. So you introduce individual phase field for each and every cell. So that's quite a lot of computational cost, but if you do it right, it's actually not that expensive. So it's so each cell is described by its own phase field, the same as I did before, but there are two additional terms in this description. This is what mathematicians use to convince people. Okay, so there is one term here which ensures that cells don't penetrate each other. So this is a penalty for penetration. So this couples phase field of different cells. So it's what it does. So if these phase fields overlap, this integral is not zero, and it penalizes the phase field. So it forces phase field to decrease. So essentially, it's like hardcore. It's not hardcore. It's softcore repulsion. So, and there is also this term. This term describes the cell-cell adhesion. So this is a really important thing too. So cells, when they interact, they of course interact mechanically. They press against each other, but they also interact biomechanically. So they develop cell-cell adhesion, which is a complex process, which involves formation of cadherin bonds, which I will be talking later, and it's effective short-range attraction. So this is described by this term. Basically, what it does. So if you have a neighboring cell, so it creates a gradient of other parameter, which forces this cell drift towards it. So this is short-term repulsion. So essentially what we do, so we solve the same equation for each cell. So equation for the order parameter, for the polarization, for the adhesion. Of course, they share the same elastic substrate. This equation is the same for all cells. But all three fields are individual for each cell. And they interact via these two terms. Okay. So this seems to be really enormous computational task, but you often can optimize it, for example, because phase fields are localized for each cell, so you can essentially zero everything outside a certain radius of the cell and reduce the number of calculations dramatically. So the first version of this thing ran about a couple of hours on a single GPU, and we had about 40, 50 cells. So now with dramatic increase in the GPU power, we can run up to 1,000 cells. So that's a significant amount. It's still not trillions, but it's for many aspects like formation of organelles, it might be sufficient.
The self-interaction term, it favors row 0, row 1.
Yeah, so when it's
self-interaction, why is this delta I?
Which one?
Yeah, no, this B, this, yeah, here.
This one.
No, not interaction, self-interaction, this one.
Oh, this is delta is a number which controls the volume of the cell. It's not delta function. It's a parameter which includes integral over cell volume. Yeah, yes. So it includes the cell volume. So each cell has its own volume. In fact, you can make it different, slightly different. Okay. And then it's coupled to adhesion dynamics and everything else. Okay, so the first thing we investigated this collision between the cells. And depending on the parameters and the shape of the cell, you have two major scenarios. The first scenario, when you have keratocyte-like cells, they collide inelastically, so like grains. So they collide, and after the collision, the orientation angle is smaller than the incoming angle. So they collide and they move together. So, and there is actually something opposite. For cells which like a fibroblast, they kind of softer and they have longer tail and they have hyperelastic collision. So the angle actually increases after the collision. So if this first scenario is favorable for the formation of tissue and collective migration, the second would favor dispersal of them. Now let me show you a few really nice movies. So these are cells in periodic boundary conditions, and they're initiated with random initial conditions, and interaction is only via collisions. So, but these are soft objects, and they do something really interesting. So they collide for some time, and after some time they all move together. They form collective migration. So this is something reminiscent of transition in the Vishik model of interacting particles, and we even calculated the other parameter, which is average velocity of all particles, and it eventually increases. It's adhesion density. Adhesion density. So each cell is deformable, so here it's about 40 cells. So we also have collective rotation. So this is one of the experimental when kidney cells. This is kidney type cells, canine cells were inoculated in a small patch, adhesive patch, and around this patch everything is not adhesive. So cells don't want to cross the boundary, and they start to rotate collectively. I will talk more about this. Apparently, it looks like solid body rotation. So at this time, when we studied it, we didn't have solid body rotation, but our cells, due to interactions, also developed this collective rotation. What is interesting, so it's you would think that adhesion, cell-cell adhesion, would promote collective migration. Apparently, it's for small density, it's not. So what? This is effect of cell-cell adhesion. Actually, when you introduce cell-cell adhesion, so they form intermittent clusters, and these clusters actually destroy collective rotation. So then, if you increase number of cells, and so you have some kind of situation which is similar to confluent layer when everything is occupied. And in this case, dynamics is interesting. So first you have some mechanical oscillations, and then cells to start to compete for voids. So they all self-propelled, they want to move, but there is a hindrance from the neighbors, and each cell this is the trajectory of the cell, they execute some sort of random motion. Okay, so now let me show you results which are not even published yet, so it's even not sent to publication. Paper is written, so we decide to explore a little bit more in detail cell biochemistry. So what I introduced so far, it was like cell machinery was described by some vector P, which is gross oversimplification. So cell machinery is more complex. In fact, it's associated with excitable network. So it's more, so it's described like some sort of inhibition activation process. So there are certain proteins which activate growth of actin filaments, and there is inhibition, and this results in formation of intracellular waves. So inside cell, you have polymerization waves. So it's action dynamics is described by a complex network. And this network is described by the most simplified description would be described only by two variables. So one is the activator and another is inhibitor, so two variables. And we use phase field model to embed excitable system, and cells now interact via excluded volume interaction and cell adhesion, but inside each cell, there is this network. So this is the work with a group of Volkan Lozart, and most of the work was done by his very capable graduate student, who is now a postdoc, Corey Herr. And so it's a little bit more complicated system, but basically it has the same components. So it has the phase field equation, but for some reason now it's not called rho, called phi. So each cell has its own phase field, and it has its own excitable network, which is described by variables Ri and Ai. And this term is responsible for the conservation of the cell volume, and this couples dynamics of the phase field with, okay, I missed A, label A, with activation field A. So when activation is created, so it stimulates protrusion of the phase field. And this term describes repulsion between the cell, the same, and cell-cell adhesion. And dynamics of activation and inhibition described by two coupled equations, partial differential equation. This is like standard reaction diffusion equation, very similar to Fitzhugh-Nagumo kind of dynamics. If you leave it alone, it will generate excitable pulses. And eta is noise. So it drives this excitable dynamics. And what we discovered is actually quite interesting. So it's I just show you snapshots of our results. So now these are self-localized clusters of the cells. The cells adhere to each other and they don't run away. And they communicate via excluded volume interaction, and nevertheless they synchronize the excitable networks. So this colors this concentration of activator. So it's you see that they execute quite complex dynamics. It's about 100 cells, and after some time they form a self-rotating cluster. And for slightly different condition, it has mechanical pulsation. So stay tuned with. I will talk more about it maybe later when we submit the paper. It's just a preview. So you see that it's form a confluent layer, but topology of the layer always changes because cells move past each other. Okay, and what is really interesting, so if you look at long-term dynamics of this excitable network, so these internal oscillators synchronize. So there is a really interesting transition which couples the onset of collective migration and synchronization of excitable networks of the cells. And this is what needs to be shown experimentally. Again, so the funding agency got across. We submitted few proposals and none of us got funded so far. We still keep trying, but once, okay, but I believe these experiments will come along eventually.
The cells would be rigid.
Yes, absolutely, yes.
But would you get the same sort of synchronization?
No, synchronization comes from very intricate and subtle phenomena from the deformation of the boundaries. So there is no signal exchange between the cells besides volume interaction. So if you make them rigid, you lose it. Yeah, sure. And there are many interesting things that happen in this system, like mechanosensitivity. And okay, yeah, you have.
One more question. So the boundaries, these are just the regions where the phase becomes small?
Yes, 1/2.
1/2.
Yeah, it's.
But the natural very
Yeah, they're very sharp. Yes, the regions these are sharp. Yes, boundaries, and it's really beautiful. So what is interesting? So you don't need any localization. So if you have enough cell adhesion, so they form these rotating clusters or migrating clusters. Okay, so if you're really excited about physical models for motility, we published a book which describes several approaches like that, and quite a few publications and more coming, in fact. So it's okay. So now let me switch gear and talk about something totally different, which was published last week, in fact. Let me stop sharing. And this one, yes, new work. Zoom. Okay, this work is about coarse-grained description of collective cell migration. So it's published last week in Communication Physics, and this work is done in collaboration with my friend and collaborator for many years, Lev Simrin from the University of San Diego. And okay, you will see. So, these phase field models are really effective for description of each and every cell, but they're exceptionally time-consuming. And if you want to understand long-term behavior and collective properties, probably it's a good idea to do some kind of coarse graining or homogenization. Mathematicians like this word more. And this can be done, and it's not trivial, but understanding collective properties would be very useful to reconcile with many experimental observations of collective cell migration. So it's I already told this. Okay, so far what I talk about is this individual. Okay, this individual cell was a cornerstone of the description, but now we have many cells, millions of cells, trillions, whatever. Okay, so cells they proliferate, so they divide. And when they divide, they often form tissues and organs. And favorite assay besides individual cell is a cell monolayer. So it's called, if there is no open space, it's called a confluent monolayer. And there are many experiments which are done with confluent monolayers. And I think the leaders in the field are people from Institute of Curie and Lado and their collaborators in Barcelona, Xavier Tripat. And they did a lot of really great work. And this is something what we focused recently. So to understand what are the collective properties of these monolayers. So confluent layers, as I mentioned, they are formed via cell division and proliferation and merging of cell clusters. And often in cell migration is suppressed in these monolayers because cells adhere strongly to each other. And they develop persistent adhesive contacts, and confluent monolayer behaves like a soft solid. And good question is, what are the properties of this solid? This is highly unusual solid. This is one of the videos, dorsal closure. So you see a lot of things happening, in fact, there. So, and many experiments demonstrated that cell tissues exhibit viscoelastic responses, and they simultaneously show liquid and solid-like behaviors. And non-trivial responses emerge when a chemical or physical stimuli are applied. And moreover, mechanical behavior may also change in time because cells can divide and die. So this is a very unusual material which may change property in time, may change topology, and respond on external stimuli. And in a few minutes, I will present you key experimental observation illustrating coexistence of solid and liquid-like behaviors. One of the most striking examples is invasion of narrow channels by migrating cells. So if you consider a fluid which invades a narrow channel, you would think about Poiseuille flow and parabolic velocity profile, right? So these experiments were performed like 10 years ago, and this cell were invading these channels, so you can see, and the velocity profile demonstrated this kind of structure. So you have almost no velocity dependence, like flat velocity profile, and then narrow boundary layers, which would consistent with solid with slightly melted boundaries. So it's for fluid, you would anticipate parabolic velocity profile. Here, your velocity profile is like plug flow. So it's strongly non-Newtonian. And confluent layer also show collective rotation. This I already mentioned. So, so if you place cell in small patch, they self-organize and rotate. It's hard to say it's solid body rotation or differential rotation. The authors argue that it's solid body rotation. So they move collectively again. So it's again, so think about it. So this is actually something very striking. So cell has well-defined velocity of migration, and collective rotation would require linearly increasing velocity. So that should be some rather intricate balance between adhesion and propulsion in order to have collective rotation. This is another experiment, which is really cool one. It's called endothelian reawakening of cell motility in confluent monolayer. This is the work was done pioneered by Roberto Scherbina. From that time, he was at University of Milan. Then he moved to University of Wien, and this is really cool experiment. So it's the confluent layer, which pretty much does nothing. So the cells are strongly in contact, was treated with a protein, endocytic protein RAB5A, which disrupt cell-cell adhesion. So cell, even they don't move, they still polarized. And if you disrupt cell-cell adhesion, they want to move, and they will move. And what they observe, the onset of large-scale migration. So the whole layer start to move collectively, and then it stops again. When cell again develops cell-cell adhesion, it stops again. It takes about like a couple of hours before it stops. And the final experiment, this is something really cool, the flow past obstacle. So this is an experiment, I don't remember, from White's group. So now cells move past obstacle. So it's like von Karman flow. So the argument from this work was that this is canine kidney cells. They flow past obstacle, and what they claim that the constitutive relation consistent with Maxwell viscoelastic model. So it's not a viscous, not a solid, but fluid, more likely. And the relaxation time of this Maxwell model would be about 70 minutes. Okay, these are experimental observation which we decide to rationalize with left simmering. We started to work last year. Actually, to obtain this result took faster than to publish this paper because reviewers had hard time to understand it. I will show why. Okay. Cell monolayers, okay, summary of experimental observation. Cell monolayers may behave like soft solids and flow like self-propelled liquids. So there is intrinsic coexistence of solid and liquid-like behavior. And they also exhibit some sort of slow glassy relaxation. There is a lot of papers about glassy-like behavior of cell monolayers, which are attributed to some intrinsic randomness, by the way. And in order to develop continuum theory, we need, if you don't really want to go deep into the biology, which is super complicated, we want to include the fundamental physical mechanism, which in this case would be self-propulsion, substrate and cell-cell adhesion, velocity alignment of neighboring cells, and contact inhibition of locomotion. The contact inhibition of locomotion means that if cells bump into each other, after some time they repolarize and go opposite directions. Okay, so in the theory, which we will essentially include all this for fundamental observations. Okay, so now this, because collective cell migration is a very popular topic, there are many models and work with mixed success. And one of the most popular models is called the active vertex model. Let's mix, and the model is cute. It's not clear what kind of relation it has to real cell monolayers. So the idea is the following. So it's people may disagree, but really it's kind of not clear what it does. So basically what it does the following. So you may have some cell center of mass, and then you draw Voronoi tessellation around them, and then you impose that the area of each Voronoi polygon is conserved as well as the perimeter. And you come up with this energy, and you minimize this energy. And then you add some sort of self-propulsion. Okay, the good question, if it really describes cell monolayers, okay, so the pictures looks kind of nice, but if it's really has anything to do with reality, it's not clear. So the model is somewhat disconnected. So it's really hard to calibrate what the forces are. So because now you have not even pairwise interaction, you have everyone interacts with everyone because of that. So it's because you minimize this thing. There are various versions of it, but it's in essence, it's always like that. So you have some sort of energy which wants to conserve perimeter and want to conserve the area. So opposite to that would be to do multi-cell phase field, which works really great. So you remember this from a few minutes ago, but this is very expensive. So this requires significant computational power. And also it does not give you direct insight what are the mechanical properties. You can measure them indirectly, but it's really hard to connect property of individual phase field to the macroscopic property of the material. Okay, there are also continuum approaches, and this is something really weird. So the most popular one, which is put forward by Julia Yeomans and her group, is to use active liquid crystals. And the motivation, I found something very unusual. So look at this. This is experimental image of a cell monolayer, and the argumentation is the following. So let's draw a nematic directory along long axis of the cell. So if cell had deformed, we say this is the nematic orientation. So you introduce the nematic field, and if you introduce the nematic field, then it should be liquid crystal. If it's a liquid crystal, so we introduce the active term, which is active stress, and solve this equation, and we believe that this will describe the cell monolayers. I find this logic somewhat twisted, so it's also not clear what is the correspondence between real cell monolayers and liquid crystals, because cells are polar, so it's not clear how you introduce liquid crystallinity. But okay, this is a quite popular approach, rather complicated in fact, but not clear. So it's, let's look at this. So do you really believe that it's a nematic liquid crystal? So I think it has nothing to, so if you take, for example, just take deformable particle, you compress them, you can always introduce nematic order parameter this way. It doesn't make it liquid crystal. Okay, so what we did, in fact, is this. So we can see we've been inspired by snow avalanches. So this is a competition for the Darwin model on the left for the most ridiculous death. So you see the avalanche going down and some idiots come up. But unfortunately, nobody was killed. Medal was not awarded. Okay, and what could be in common with the cell monolayers? So if you think about it, practically nothing is in common. So it's granular avalanches are driven by friction and gravity, and cells are self-propelled. The scale is very different from 100 meters to 100 microns. Time scale is different. So one of the most striking differences in granular system, you don't have tensile stresses, you don't have adhesion. Here you have elastic solid. And these are inertia driven, this is overdamp, these are rigid particles, this is deformable particles. So essentially, the right answer would be there is no similarity between these two phenomena. But this answer probably would satisfy a layman, but not a curious scientist. So it's, if you examine it once again, so you see some similarity. Both granular system and thermal arrays, they exhibit coexistence of solid-like and liquid-like behaviors. So avalanches, they present in the, before they become avalanches, they present in the solid form, and then small perturbation converts them into a liquid, granular liquid. And transition between liquid and solid is governed by shear stress, which itself depends on the configuration. And like almost 20 years ago with Lev Sivnik, we developed a partial fluidization approach, which captures solid-like transition in granular systems. And we actually use this approach to describe these cell monolayers. So in essence, the granular, for granular avalanches, we use the following approach. So we stipulated that a granular material like sand or snow can be present in two forms. This is the same material, but the physical state is different. Liquid-like when it flows and solid-like. And in order to bridge these two states, we introduce another parameter called rho, surprisingly, which bridges these two behaviors. And other parameter is, say, 1 for solid and 0 for a liquid. And then we use the same other parameter equation to describe the transition, where the parameter delta, the same which was for the phase field, also phase field description, depends on the shear stress, while Morculon failure condition. And this actually was quite successful description, and now it's used in granular matter. We published quite a number of papers about it. So the most recent one was Review of Modern Physics and a monograph granular patterns. And then we examined all these experiments, which I showed you. So we concluded that actually we can use this analogy. We can use this other parameter description to reconcile liquid-like and solid-like behavior of cell monolayers. So our approach is the cell monolayer is viscoelastic but highly unusual material. And we use fundamental laws of physics, which is really hard to break, like momentum conservation and experimental observations and most real biological interactions. So the model in some self is minimal. So it's, and the primary innovation is to use the order parameter to bridge solid-like and liquid-like behaviors. And the first equation is a momentum conservation, so which is rather universal. So we say that the cell monolayer, which is adherent to the substrate, satisfies the momentum conservation, which is the balance of forces. So this is the drag. V is the velocity of the monolayer. Fp is the propulsion. So cells impose force, which is proportional to self-polarization with the strength F. Basically, this is biology included here. So this is a tendency of cells to propel. So they impose the force. And this is a total stress tensor, which includes the internal forces. And to make life slightly easier, we assume that the monolayer is incompressible. Okay, so we could make it compressible, which doesn't change much, or we can even include proliferation. And then it would be extra terms here, but this would make life significantly more complex without really adding something fundamentally different. So for the moment, we assume that the monolayer is incompressible. Okay, then the stress tensor can be written in the following form. It could be a combination of a deviatoric viscoelastic stress tensor and isotropic pressure. Okay, so here is something really subtle. So in fluid dynamics, in compressible fluid dynamics, pressure is irrelevant. right? It's a Lagrange multiplier. You can define pressure up to a constant. Not here. Here, of course, from the fluid mechanics point of view, pressure is defined up to a constant, but pressure is internal variable, and it controls the cell state. For example, if you increase pressure, cells stop to divide, or it also changes the cell-cell adhesion. So the pressure is an important parameter, and it will be important parameter. So it's if boundary conditions are periodic, so it's effective boundary conditions is that average pressure should be imposed. It's a control parameter. It's not arbitrary constant. Okay, so and then we come to the state equation of state, so Maxwell model, and the Maxwell model is described in the following form. It's similar to Oldroyd B model, but there is one important caveat, which I will explain. So it has, this is rate of strain tensor, and this is the derivative of the stress tensor. And mu E is elasticity, and mu is the viscosity. And primary innovation here is the dependence of the relaxation time on the order parameter. So this was not done before. So this is highly unusual viscoelastic solid. So what do we have? So if we consider the order parameter, or the parameter tends to 1, so we can drop this term, and then we have the derivative of the stress tensor equals to the rate of strain. And if we integrate once, we obtain the elasticity relation. So the stress tensor is proportional to the displacement. So if order parameter tends to 0, then we have more or less standard Maxwell liquid. Okay, there is some important caveat here too. So in this kind of field, everything is tricky. So if you look at many publications in this field, so everyone wants, especially at the Oldroyd B model, so this material derivative actually has more terms. So in principle, in Oldroyd B model, you should have more terms, not just advection by the velocity, but also these terms, which describe alignment of the material along the principal axis of the tensor and rotation. And but this objective representation of the tensor requires frame invariant representation. So it would be valid if you work with bulk material, but in our case, cell monolayer adhere to the substrate, and we have preferred frame. So we argue that these terms should not really appear, which if you look at literature in this active matter, it's depending on the author. Some people include them, some don't. So we argue that they shouldn't be there, in fact. But self-propulsion actually imposes this term, because cells are self-propelled, you still have advection. Okay, so and order parameter captures important physics. So it captures variable relaxation time, and it's related to the following phenomenon. So we define the order parameter as a fraction of adhesive bonds between the cells. So the cells may have adhesive contacts and may have sliding contacts. And when cells adhere to each other, so they do it in very intricate way again. So via these crosslinks, which are called cadherin proteins. And cadherin proteins, they crosslink actin filaments of different cells through the cell membrane. And this interaction is called catch-bond interaction. So it's this strength of this interaction increases with the applied stress. So it's catch-bond interaction. However, cross-linking requires very significant time. So it's hours. If cells slide past each other for the same force, they actually don't form these bonds. So bonds, to form these bonds require time. And if they initially slide, the bonds are not formed. So as a result, you have two different states, which are captured by the order parameter. So for this situation, the order if all the bonds are adhesive, the order parameter will be 1. And if all the bonds are sliding, it will be 0. Okay, and then we introduce the order parameter, which we already presented in previous lectures. It's the same game. So we introduce the fluidization of the parameters described by this reaction diffusion equation or the Ginzburg-Landau equation, which has bistable potential. It has potential with two minima, so this you can introduce as free energy. And this free energy, fictional free energy, has two minima. One minima corresponds to solid, 1, and to liquid, 0. And the relative depth of this minima controlled by the parameter delta. And parameter delta, in turn, depends on the applied shear stress. So it's somewhat similar to what I did for individual cells, but now physics is different. And okay, this we don't need. And now we need the parameter delta, which depends on the shear stress, can be introduced via classical mechanical Moore-Coulomb failure criterion. So understand it is, you can imagine this block on the inclined plane with the friction. If the angle of the plane exceeds certain critical angle, block starts to slide down. So this is mechanical failure. Basically, it simulates the same. So if the shear stress becomes larger than adhesive stress plus internal friction coefficient times normal stress, the material fails. And you can calculate this failure criterion using our notations. So you need to calculate magnitude of the shear stress, which is given here, divided by the normal stress. And normal stress in our notation is P, but P is not arbitrary. It's actually described as, it's in fact, is determined by the boundary conditions. And the parameter delta naught describes this analog of analog of cohesive interaction, cohesive strength. So this is a parameter which couples the dynamics of the order parameter to the mechanical stresses. And finally, we have a last equation. Again, it looks like the Ginzburg-Landau equation for the cell polarization. So the cells spontaneously, so if they're polarized, they want to keep polarization. And the structure of this equation is quite simple. So it's people working on active matter, of course, know it very well. It's classic equation for polar active matter. So this includes advection, so it couples to the velocity. This term counts for cell-cell alignment. So if you have neighboring cells, the orientation wants to be the same. So for example, if you have cells which are aligned in different directions, the diffusion would make them align in the same direction. This is like V-shaped interaction between the neighbors. These two terms, they maintain the absolute value of P close to 1. This is due to cell metabolism. So cell use nutrients and they keep pushing, they're alive, and they maintain polarized states. They exert the force all the time. And the last term couples to the pressure gradient. It accounts for the contact inhibition of locomotion. Basically, it says the following. So if there is a gradient of pressure, cell doesn't want to go this way. It wants to go opposite way. And this is introduced, so it basically aligns polarization against the gradient of pressure, but this P, nabla P, introduced such a way that gradient of pressure doesn't modify the magnitude of polarization. So if you just introduce gradient of P, so it will change the magnitude of polarization. So this term, this is like a Landau-Lifshitz term in magnetization equation, ensures that gradient of P doesn't change magnitude of polarization. So these are three fundamental biological interactions. So cells want to align with the neighbors, maintain polarization, and migrate against the gradient of pressure. That's about it. So the model is relatively simple. And first, you can examine it in one dimension. So like flow in the channel. So in this situation, you can simplify it quite a bit. Polarization maintains only one component along the channel. velocity has only one component, Vx, which depends on coordinate across the channel, Vy. And there is only one component of the shear stress, sigma xy, and pressure is constant, equals to P naught. So if you do all these simplifications, you obtain essentially two partial differential equations. So one equation for the shear stress, so you can plug this in, and one equation for the order parameter. And they coupled via this term, this Moore-Coulomb-Feilly criterion. So this equation is easy to solve, MATLAB or whatever. And what it does actually makes perfect sense. Basically, it tells you that if there is monal array which flying in thin channel, so the order parameter is depressed near the walls, so there is partial fluidization, so it flows like fluid, and then it acquires value of 1. So it's inside the channel, it's like a solid. And the velocity profile also has this plug flow. So it's essentially constant in the middle of the channel, and then it rapidly decreases at the edges of the channel. And this is consistent with what was measured in experiments. So experiment also demonstrated that there is this plug flow, very different from the Poisson flow. So it's like essentially you can interpret this flow of the cell. It's like molten solid. So it's solid in the center and molten fluid near the edges due to shear. You can do quite a lot of analytical stuff. I don't go into detail. So you can obtain the phase diagram analytically for most of the cases. And it shows bistable behavior, so with hysteresis. So you have two states which coexist, depending on initial condition. And in this is the length of the channel, and this is normalized propulsion force. And in this area, depending on the coefficient, you have coexistence on liquid and solid phases. And even the profile of the order parameter you can calculate analytically in this limit. Okay, so interesting question happens, what if you have two-dimensional situation? So you have channel, but in 2D. Then, okay, then you need to do numerics. And so numerics actually is much simpler than what I showed you before. So it runs on this machine, on this notebook. So most of the simulations we did in periodic boundary conditions using quasi-spectral split-step method based on the FFT. And most of the simulations we use 1024 by 1024 FFT harmonics. And to solve incompressibility conditions, we use stream function formulation. So this is what I say, if you do it compressible, that's life becomes certainly much harder without bringing much, and runs on MATLAB. So in fact, if you're interested, so it's in our paper, we supply the MATLAB script. So you can run it on your notebook. It runs few minutes, very fast. The code, it's about 100 lines only, so it's very efficient. Okay, so this is the flow inside the channel in 2D. Okay, so let me explain why you will see two channels, one going this way, one going this way. This is because we use stream function formulation, and in periodic boundary conditions, you may not have velocity going in one way, because it would violate, make stream function non-periodic. So you need to have two channels with opposite flow because of periodicity of the stream function. This is some technical stuff. Okay, so now first we did simulations for relatively narrow channels. And white arrows are velocity vectors, and green arrows are polarization vectors. And red, in all other videos, would correspond to order parameter 1, and blue to order parameter 0. So you see that starting from, I can play it again. So we started from random initial conditions, and after some time, the flow self-organized in such a way that all cells go in the same direction, and velocity and polarization are strongly aligned. Again, because of this periodicity condition, you need two channels. So now what happens if you increase the width of the channel? So it's make it wider. So there is another transition. So it's so now the same situation, but wider channel. And you see the flow never settles down. So in wider channels, the cells move in somewhat complicated fashion. So they have some vortices and this kind of flow has also been observed. Okay, so it's we did not do it, but I think it's certainly possible to do to calculate analytically this transition, at which critical lengths you would have this transition from steady flow to this unsteady flow. You can do linear stability analysis and you can calculate it. It's kind of involved. Okay. So so now let's see what happens in large systems. So it's not so this system was confined in this channel. So now what happens if your system is large? And what we observe, so this is a simulation of this relatively large system and periodic boundary conditions. Again, in this white arrow shows velocity and black arrow shows the polarization. And this bright yellow corresponds to the order parameter 1 to solid and blue to liquid. And this panel shows the shear stress and this panel shows the rate of strain. So for generic initial conditions, system, depending on the parameters again, system evolves towards coexistence of liquid and solid. So you have large islands of solid, which actually move. They are not stationary, they move as whole. And they're separated by these channels of liquid, and which lubricate them. So they can slide past each other because of that. And what is remarkable, so this contour lines corresponds to the order parameter 0.5. So they confine fluid. So fluid is inside, as you can see. And what you can see actually makes a lot of sense. So the rate of strain is localized in liquid and practically absent in solid. So solid may not sustain rate of strain. It may sustain strain only, right? Because it will react elastically, but rate of strain only localized in the liquid. So you have this coexistence of solid and liquids, and it's never settles down. So it evolves this way, depending on the parameters. So the question is if this been observed experimentally. That's a good question. I'm not sure. So there is a paper which shows something similar, but interpretation is totally different. So there's a paper from Dave White's group and his Chinese collaborators and they claim that what they observed is non-Gaussian statistics, like Q Gaussian statistics, which I'm not sure that it's really a case. So it would be interesting to reinterpret it in terms of this approach.
We're talking about this reawakening in this Roberto's.
I will talk more about it. Yeah, I will talk more about it, yes. And this would be kind of reawakening, right? But the opposite. So you start with the solid and then you break the adhesive contacts, right? Okay, so this is really remarkable. So this actually can be, again, I don't think Benoit Lado or Xavier Tripat really look at this, but they could, in fact. So localization of the strain. This would be one of the hallmark of this transition.
This happening at 1,000?
No, I will show the video. No, it's happening. This is the video, yes. So we start with fluid state, so it's no adhesive contacts, and then they develop. So this is how it evolves. So red is solid. And you see there are solid islands, and they actually evolve, and quite complex evolution.
You have introduced contact inhibition of motion.
Yeah.
How relevant is that mechanism to observe this behavior?
Hard to say. It's not super relevant. It may have some features, but I think it would exist without it. So the problem is that in solid you would have contact inhibition of locomotion provides coupling between stress and polarization. If you don't have it, there will be no coupling. And I believe there is coupling between this, right? So there is another coupling, which we didn't include, by the way. So there is also, people argue that there is a phenomenon called politotaxis. So orientation of polarization along major axis of the stress tensor. This happens to be totally irrelevant. So we tried and it didn't show much effect. So this makes life, of course, much harder, but we didn't see much difference. Okay, so you see that there is really complex dynamics of solid phase and liquid phase. So solid phase expands, but still there are these lubrication layers which separate them. And there is a parameter here. So if let me show you what it seeds. Okay. Yes, this parameter. We call it stress susceptibility. Basically, it measures how sensitive this transition is to the applied shear stress. So if this parameter is small, there is very little susceptibility, and you will favor solid. If it's large, you will favor liquid. Okay. Yes. So we did analysis for different values of the parameters, this kappa, stress susceptibility, and what phi is a filling fraction of solid. And if stress susceptibility is small, so it's very hard to melt. So essentially, the system evolves towards solid very fast. So it's for some intermediate values, you have fluid and solid coexistence. So you have some lubrication layers of fluid. And for large values of kappa, everything melts. Phi goes to 0. And this shows velocity, average velocity as function of time for different stress susceptibilities. And for liquid-like states, the velocity is approaching maximum possible velocity, which is F is the strength of polarization, is close to 1. And for solid-like, velocity goes to 0. But it goes to 0 in a very intricate way. It is not monotonous. You see already some oscillations here. So we decide to investigate what is really going on. And okay, so this video shows you relaxation of the solid phase. The relaxation of the solid phase happens to be highly non-trivial, which features sort of glassy transition. But again, here there is no, I have like half an hour, right? I probably finish even earlier. So in previous work, glassy behavior was attributed to intrinsic randomness of the monolayer, which is maybe a reasonable idea, but attribute everything to randomness, that's basically says that we don't know what is it, right? So what is shown here on this panel, okay, let me, so in this panel, we'll show maximal velocity, magnitude of the velocity, not the other parameter, but the magnitude of the velocity. And on the other panel, it shows the shear stress. And essentially, the other parameter is very close to 1. And what you see is the maximal velocity develops these hot zones. They appear spontaneously and disappear. So actually, relaxation, so other parameters close to 1, velocity decreases. See that velocity magnitude decreases all the time, but it's highly non-monotonous. There is activation of some hot spots. And relaxation happens via formation of these hot spots, like in glassy systems. So if this is the case, hard to say, so the system shows it. And what is also remarkable is that polarization and velocity are totally decorrelated. So this is a 3D diagram, this X, Y, and T, and dots are position of these hot spots, the maximal of the velocity. And you see they actually have very complex dynamics. They drift, they spin, and they appear and disappear. So it's this something like that you would understand in glassy system, but the origin of this glassy-like behavior is purely deterministic. It's not related to randomness or heterogeneity. And this plot shows velocity relaxation. And so red line is average velocity, but black line is maximum velocity. So average velocity shows some decay, but maximum velocity shows oscillation. So basically it corresponds to formation of these hotspots. When hotspot is formed, so locally you have relatively high velocity, and then it disappears. And again, polarization and velocity in this solid state completely decorrelated. And this you can understand. So in order to maintain zero velocity, cells want to push. They have to push, velocity and polarization should be decorrelated. If they aligned, system will move, right? And if it doesn't move, the velocity and polarization should have roughly in average opposite directions. Okay, so we introduce the velocity polarization correlation coefficient. So if they would be totally aligned, it will be 1. If anti-aligned, it will be -1, otherwise close to 0. So this is velocity and polarization. And for the solid, essentially correlation coefficient goes to 0. So these two fields apparently decorrelated, and which is kind of surprising. But and in solid phase or partially fluidized phase, they almost immediately correlated, very close to 1. So if system moves and melts just a little bit, they become immediately correlated. But when it stops, they lose correlation. Okay, then it's next thing we examined the onset of solid body rotation. And indeed, for small patches like that, so this is adhesive patch, and everywhere here is non-adhesive. For small susceptibility to the stress, we observe solid body rotation, which is even surprising in this situation. Velocity and polarization are decorrelated as for the solid state. As you can see that arrows point in different direction, but the velocity profile is very close to linear. So if we increase the stress susceptibility coefficient just a little more, significantly, and allow system to melt, then we have differential rotation, and velocity and orientation becomes immediately correlated. So let me show you a couple of videos. This is the onset of solid body rotation, and happens to be quite non-trivial. Quite a lot of things going on there, but eventually that's established. Polarization and velocity oscillate, but in average, you have solid body rotation. And this would be consistent with this video, the onset of solid body rotations in small patches. So in this kind of study, so we applied effective sleep condition on the surface of the patch. So it can sleep freely, like an experiment. So now if you increase the value of susceptibility coefficient, then it melts immediately in the center, somewhere here. And you have differential rotation. Okay. Okay, now something really cool. So this framework allows us to investigate response on perturbations in space and time. And this is Ignacio question about Roberto's experiment. So we can do this. So let's see what happens. So, but we can do it even more in a different way. So what we examined is the following. So we prepared a solid state. So this system evolves towards solid. And then we introduce perturbation somewhere here, localized perturbation. So we can disrupt cell-cell adhesion, or we can disrupt cell polarization. We can do different things. Actually, response will be very different. In the first simulation, we disrupt cell-cell adhesion. So it's basically, if we disrupt cell adhesions, the monomer will immediately melt. And then it solidifies. So what you will see in this video, so first everything becomes red, so it's solid. And then we, in the center of the domain, it becomes blue. So we melt it So we disrupt cell-cell adhesion by intentionally setting order parameter to 0 in this small domain. So it's solidifies, it becomes, so velocity decreases, and then we melt it. And then it shrinks again. And system actually keeps relaxing. And response of this perturbation is somewhat localized. It repairs this damage very fast. See? There is no global rearrangement. So this essentially, this perturbation is, response to this perturbation rather local. And now we do something different. Now we disrupt polarization. So in this small circle, we set polarization to 0. And this triggers something totally different. Because this is a solid, or in solid, there is intricate balance between polarization and shear stress, right? So because system doesn't move, the shear stress should balance propulsion. So now we disrupt polarization in this area, and this global balance is destroyed. And systems start to rearrange globally. So response is more dramatic and more long-term. So again, so it doesn't melt. So now, yes, we introduce perturbation. I see you now, you have rearrangement everywhere. Somewhere here too, yes. Okay, so this is can be summarized here. This is treatment window. So this is relaxation of velocity and the order parameter, and this is treatment window where the either 0 cell-cell adhesion in this window or 0 polarization. And then we see the response. So if we 0 the cell-cell adhesion, so we have initial increase of the velocity, but then it relaxes rather fast. So what is interesting, so if we disrupt polarization, so we have some sort of echo effect. Because of global rearrangement, so the stress, the force balance, global force balance changes, and you have echo effect. So you have first response right after the perturbation. So you have increase of the velocity, and then after some velocity increases again. So there is secondary and tertiary peaks. Okay, now let me come to conclusion. Okay, so we developed self-consistent description of solid-liquid transition confluent monolayers. We reproduced experimental phenomenology like plug flow, solid body rotation, and slow glassy relaxation. And there are testable predictions for experiment with respect to localized spatial temporal perturbations. And the model can be readily extended to 3 dimensions: curved substrate, cell signaling, hemataxis, and cell proliferation. That would be quite straightforward. And parameters of the model can be extracted from the particle model. So I don't think we want to use vertex model. I don't believe, because it's too unpredictable. So now with left sim, we're trying to develop analog of the vertex model, but more driven physically, like particle-based when you have real particles, and it's kind of working, but we keep it's. So we can measure the order parameter by counting the adhesive contacts between the cells. So this is red is the order parameter, V is the velocity profile. So if you now introduce adhesive contacts and sliding contact, you can measure the fraction. You can see it kind of shows similar behavior. So next thing what we want to do, we want to calibrate the parameters of the model from this kind of particle-based simulations. Okay, so it would be really great to calibrate also from experiment, but it probably doesn't happen anytime soon. Okay, thank you very much, and questions?
Related to this connection to the vertex model, I understand your comments, but I wanted to ask, on the one hand, since you have these particle-based models, whether you can compare to what vertex predicts, that is more, I mean, so, mathematics is very much about this T events, and I don't know how much you can capture effectively by particle.
No, this is not vertex model, so it looks like
No. Yeah.
but it's,
So
it's
what
really
is that model then? What is that?
It's
behind that
okay.
model?
So this model is very simple. So we have particles with repulsion forces and with adhesive forces, and we performed this Voronoi tessellation, and for each edge of the Voronoi tessellation, we assign some adhesive contacts, and these adhesive contacts have this catch bond dynamics. So it's different from vertex model in this respect, because we try to use vertex model, but it doesn't show this kind of behavior which I mentioned. So it doesn't show coexistence.
Okay.
Any other question?
I just have a question. So there is a parameter delta. So I wonder it should be between 0 and 1, but in your
Parameter delta or parameter rho?
delta, this delta control,
It's not between 0 and 1. It may change wide range.
It's so
It's larger than, it's slightly negative because we have minus delta naught, but it may change to whatever. It can be large and positive.
So in this case, what is the interface between these two phases?
It controls the movement of the interface, but if it becomes large, everything melts to 0. So if delta is large, so it's 0. Rho is 0, so you don't have any other solution. So rho 0 becomes global attractor. So it's basically any shear stress melts solid.
Just curious if the fluid tumor better for the patient or the solid tumor?
Could you please repeat?
What is more dangerous for the patient? Fluid tumor or solid tumor?
I think fluid is more dangerous because it proliferates.
So
as long as solid, it doesn't go anywhere, right?
It depends.
It's only yeah, but it's fluid. These clusters can detach and go.
Is it possible to control the direction when there is the two of them exist?
Yes, to make it more solid, for example, you need to increase cell-cell adhesion. So cell-cell adhesion. If it's possible to do, it's a good question. So I know that it's possible to decrease it. Increasing it might be harder, but at least you should prevent to decrease cell adhesion.
Okay. In your beginning models, did you consider cell-cell signal?
No, not in this model. So it can be introduced, sure. So it's no, but it will be additional field. Yes, it is important. Yes, for tumors, of course, it's very important. Yeah, but not in this model. So this is depends. Mechanical property probably not too much, but if you self-signaling, so you may coerce the cells move in a certain direction. But this would be straightforward to introduce by introducing additional chemical field and couple it to, say, polarization.
Okay, so if there is no other question, let's close the session for today. Okay, thanks.